Well, today we're going to get an opportunity to look at how we smell. Well, let me say that again. Not how we smell, but how we're able to smell. And the person who's going to guide us is Dr. Charles Greer. And we're very privileged to have him because he's a professor of neuroscience. In addition, he's also a Harley-Davidson man. And today he'll be talking about what's cleverly titled Scents and Sensibilities, Molecular Mechanisms of Olfaction. So please, let's welcome him to the podium.
Thank you for the introduction. Can you all hear me okay? It's all right? Okay. So first, it's a pleasure to be here with you today. Second, I'm happy to entertain questions at any point. So if we're moving along and there's an issue that comes up that you'd like to explore in more detail, I'm happy to do that. So what am I? I'm a neuroscientist. I'm interested in studying how the brain is organized, and by understanding how it's organized, hopefully understanding a little bit better how it works. Now, the brain as a whole is big and complex, and it's got millions of parts to it. So it's hard to study the brain in its entirety at any given point. So what many of us do, many of us working in the field of the neurosciences, do instead is we pick out a small part of the brain, in my case the olfactory system, and we decide to study that in detail. And we look for the principles that govern the way that structure functions, how it interacts with other parts of the brain. And then as we understand that more fully, we try to take the lessons that we've learned and extrapolate and then look at another part of the brain. So I might begin with the visual system and begin with the olfactory system, take what I learned from the olfactory system, and then look at the visual system and say, well, how do the principles that underlie structure and function in smell, how do they apply to the way visual information is processed? It's like building a house. I'm going to start with one room, and then I'm going to move on to the next room. So that's one of the reasons that we study the olfactory system. The other reason for studying the olfactory system is it plays an important role in our daily lives. You don't recognize it so much. I will talk in more detail about some of the behaviors that are mediated by the olfactory system as we go on. But suffice it to say that your ability to appreciate food, your interactions with your friends and your family and the pets that may be part of your lives are all mediated or strongly influenced by the olfactory system. So there are not many well-defined diseases that are associated with the olfactory system. Anosmia in general defines a complete loss of the sense of smell. It can be either complete or partial. Imagine, if you will, the inability to smell pizza. This happens to some people. They can't smell pizza anymore. God, how dreadful. Pizza is my favorite food in the world. Not being able to smell it would be terrible. Other people have a complete loss of smell. This happens particularly in the aged. If you talk to your grandparents or your great-grandparents, they may tell you that their sense of smell is not as good as it used to be. We don't understand fully the mechanisms that may be responsible for that yet, but we do recognize it happens. Parosmia are changes in the way the olfactory system functions. This is particularly evident in women as they become pregnant. Will often find odors that previously had been appealing to them, the smell of ice cream or a particular perfume or a particular kind of food. But as the pregnancy proceeds, the smell now becomes extremely offensive. And this is a parosmia, a change in sensitivity or a change in the individual's response or percept, their recognition of the odor. Other than that, I think probably one of the better known diseases is rhinotoxomania. Now, many of you may recognize this. It's characterized by the insertion of a single digit into the nose with a vigorous movement of the finger after insertion. So if any of your teachers give you a problem in class, just say, "It's not my fault. I've got rhinotoxomania." So let's talk in a little more detail about the olfactory system. First, it's a chemical detector. Your auditory system detects changes in pressure that we call sound. Your visual system detects changes in light or differences in light levels that lead to vision. Somatosensory, we feel pressure and pricks and so on and so forth on our digits and elsewhere on our body. The olfactory system is specialized as a chemical detector, molecules that are floating around in the environment. The molecular identity of an odor, a pizza versus a pork roast versus a cob of corn and so on and so forth, occurs in a combinatorial way. What do I mean by that? When you smell coffee in the morning, or if you smelled the coffee that was in the pot out here, It's not just one smell. It's many, many, many different smells that have come together that result in what you recognize as the smell of coffee. And in the case of coffee, we know that it's more than 100 different molecules that result in what we call a coffee smell. So it's a combinatorial code. You combine all these different molecules, and that combination then results in the smell. It's like a pizza. You get a combination pizza with lots of stuff on it, pepperoni, sausage, peppers, and so on and so forth. That has one kind of smell. If you get just a cheese pizza with a little bit of tomato sauce, that has a different kind of smell. So as we combine the things together in a combinatorial code, we have the identity of the odor that we're looking at. The molecular specificity, or that odor code, begins with the expression of a receptor in the membranes of the sensory neurons that are found inside our noses. And we're going to take a look at those in a little more detail in a few minutes. Each sensory neuron in the nose, and there are millions of them, expresses only one kind of receptor. So there's not a sensory neuron in the nose for the combination pizza, and there's not a sensory neuron in the nose for coffee. Rather, they're individual receptors that look at just small components that will be part of the combinatorial code. The axons from the nose go into the brain, where they make synapses in central areas of the brain and then continue on up. And we'll look at what those central areas look like. One of the unique features of the olfactory system, which differentiates it from almost any other sensory system in the human, is that it has the capacity to regenerate. Right now, today, if you sustain an injury to your spinal cord and your legs are paralyzed, your ability to walk in the future is severely limited. You may not be able to walk at all because the nerves in your spinal cord are unable to regenerate. That's not true of the olfactory system. The olfactory system regenerates and reorganizes itself on a daily basis. It begins when we're very young, and it continues through the remainder of our life. And in fact, that's another reason to study the olfactory system as a model. We can look at the principles that enable the olfactory system to regenerate on a regular basis, take those principles and apply them to the spinal cord and ask if we can improve regeneration by taking features of the olfactory system and putting them in the spinal cord. And we're not going to talk about it today, but in fact, it does happen. We can take components of the olfactory system, put them into the injured spinal cord, and increase the probability of regeneration occurring. So lots and lots of reasons to study the olfactory system, not the least of which, as I've already mentioned, olfaction affects behavior, and we'll come back to that repeatedly. So what does the olfactory system look like? This is a cross-section of the head. You see the nose there, the lips down there, the chin, the forehead. This is the eye. The olfactory bulb is located immediately above the nose. It's right between your eyes. Within your nose are the sensory neurons. You see them shown here with these long green lines. They move from the nose up into the first part of the brain, which is called the olfactory bulb. We'll look at that in more detail. And then from the olfactory bulb, information goes back to the rest of cortex. And I want to draw a sharp distinction between the olfactory system, shown here, and the gustatory or the taste system, shown down here. They do share some common principles in that they're both chemical detectors, but are otherwise quite different from each other. And in fact, at the end of what we have, 3 hours this morning? Yeah? So at the end of the 3 hours, we'll do an experiment in which we'll distinguish between the role of smell and the role of the gustatory or taste system in your ability to recognize and appreciate flavors. So this is what your head looks like. Hopefully it doesn't look like that today because there's no brain up here. This is a cross-section. Here's the nose. This is the external nares. This is the part of your nose right here where you would pick your finger if you had rhinotolexomania. This is the olfactory epithelium. And these folds that you see in the tissue here help to warm the air and also help to moisturize the air. The olfactory epithelium itself is located just here in the dorsal recess. And when I say olfactory epithelium, what I mean is that part of the nose that has the sensory neurons within it. This is the cranial cavity the brain is missing. This is the sphenoid sinus. This is one of the sinuses that can get clogged when you get sick. And you see the surface of the tongue right down here, and this is part of the throat. We can't do most of our experiments in humans. We have to look for other models in which we can carry out our experiments. And most of us use the rodent or mouse olfactory system as a model. Turns out that the principles of organization, the kinds of cells that are found there, and the way the system functions is virtually identical or indistinguishable from what occurs in the human. So So we can use the rodent or another vertebrate model, another mammalian model, to extrapolate back to the human and understand what happens. So I showed you here that the olfactory epithelium was located there and that the brain was just up there. In the rodent, the olfactory epithelium is here, again shown in green, and that the olfactory bulb, the first part of the brain that receives olfactory information, is located just here. On the right, that organization is shown schematically. You can see the epithelium at the top, and I'm going to look at each of these cells in detail with you. And then at the bottom, you see the olfactory bulb, where the processes of the sensory neurons that are shown in green and blue come into the olfactory bulb, where they then make contact with central components where odor representation is finally going to occur. As an introduction, the odor molecules will come into the nose, shown up here at the top. They're going to interact with a particular detector, the receptor for particular molecules, which will be expressed on the surface of the sensory neurons, the blue and green cells. And then those cells will take that information, they'll pass it into the brain, where they're going to make a synapse or a functional contact with other neurons that will take that information out to higher regions of the brain. These regions where those contacts will occur are called glomeruli. And you'll see that the glomeruli at least contribute to the coding of olfactory information, the coding of odor information. We'll find that one glomerulus is going to detect a particular chemical feature, another glomerulus, another chemical feature, and so on and so forth as we go through all of the glomeruli. So we can now look at the olfactory epithelium in great detail. It's about the thickness of your fingernail, not the width of your fingernail, but just the thickness of your fingernail. So it's about 500 microns. And it has three primary populations of cells, just three. The first population, and the most important one, or the most exciting one, are the olfactory sensory neurons themselves. Each of you has about 1 million to 5 million sensory neurons on each side of your nose. And they divide up into many, many different populations. The population that they are part of is going to reflect the odor receptor, the molecular receptor that they express, and we'll look at those receptors in more detail in a minute. These cells are called bipolar cells. That means they have two poles to them. The first pole is an apical branch. It's called a dendrite that extends to the surface of the nose. If you could get your finger up there far enough, you'd be able to touch this part. Don't try. It's a bad thing to do. The second pole is called the axon, and the axon takes the information that is initially detected by the apical portion, the dendrite, and passes it into the central nervous system where the synapses occur. Now, the olfactory sensory neurons are a bit more complex. As they reach the surface of the olfactory epithelium, shown just here, there's a slight enlargement. It's called a dendritic knob, and we'll see that in a bit more detail. But more importantly, there are these long processes coming off. They're called cilia. Now, if you play baseball, if you went out to play baseball and you tried to catch the baseball just by holding up your hand cupped like that, it would be a real problem. You're probably not going to be very effective at catching the baseball. But if you put a baseball mitt on and you have the fingers extended and the fingers enlarged a bit, now you're going to have a much bigger field or a much bigger receptive area into which you can try to get that ball to go so that you can catch it. Well, that's what's happening up here with these cilia. We're going to see later on that odors come into this area, and we want to maximize the likelihood. We want to maximize the probability that when an odorant comes in, it's going to bind with or interact or bump off of these ciliary processes. So they increase the amount of surface area. They increase the representation of the sensory neuron at the surface of the epithelium, and in doing so, they increase the likelihood or the probability that an odorant will be detected by a cell once it comes into the nose. Yeah? This is a single cell, this is a single cell, this and this. Anywhere from 1 million to 5 or so million on each side of your nose. The second population of cells are called sustentacular or supporting cells. They're shown in green here. We think that they help to detoxify the nose. The nose is a nasty, nasty place, right? Get all kinds of stuff going up there. Dust, dirt, bugs, that pee you stuffed up there when you were a little kid, and so on and so forth. We think that the sustentacular cells help remove and detoxify the environment. And we also think they help remove odorants from the environment. Now, if you take a quarter and you put it on your forearm and you let it sit there for a while, when you first put it on, you can feel it quite distinctly. You can describe its shape and its weight and so on. After it has been there for a short period of time, you can no longer detect it. It's a process that's called adaptation, and all of our sensory systems show adaptation, including the olfactory system. So if we bring an odorant into this environment and just leave it there, after a while, these cells will no longer respond. They'll be saturated, they'll be adapted, there's no response. So we need to remove the odorants from the environment, and we think the sustentacular cells contribute to that process. Now, the third population of cells are down here at the base. They're called basal cells, or in more contemporary terms, stem cells. They're perhaps among the most interesting cells that are found within the olfactory epithelium. Now, I mentioned that the epithelium continues to be regenerated throughout life, and it's the division of these basal cells or stem cells that's responsible for that. I'll show you in a little while that as one of these cells dies, the basal cell or the stem cell will divide and it'll replace it, and then allow the daughter cell or the replaced cell to differentiate effectively and send its axon or reestablish a connection with the central nervous system. Now, we've talked about mucus. So in talking about mucus at this point, it's also important to understand where boogers come from. So I told you the nose is a toxic environment. There's lots of dirt in there. It goes in and interacts with the mucus. You know what mucus is like. You've all had to blow your nose from time to time. So you know that it's kind of sticky and gooey. So these little bits of dirt get in and they clump together, and when they clump sufficiently, they produce a booger. So they're really dirt. So even if you take them and line them up neatly, you know, on the desk or the table next to your bed, they're still dirty. So you really need to stop doing that if you're still persisting in this behavior. So the other reason it's good to work with the olfactory system is there are, you know, these little things about it that are kind of amusing from time to time. So coming into the nasal cavity will be dust, dirt, toxins of various sorts. This sensory neuron is a neuron. It's got a very delicate membrane. It's not able to sustain its good health with a repeated exposure to the dust, dirt, and toxins. So what happens is they act on the sensory neurons and they kill them. They die. In order for olfaction to continue then, they have to be replaced, and the way they're replaced is division of the basal cell down at the bottom, now shown in pink, which gives rise to a new daughter cell which differentiates and sends its axon out to the brain. So this is the process of regeneration in the olfactory system. The sensory neurons die as a result of exposure to toxins and dirt from the environment. If you work in a coal mine, for example, your olfactory epithelium is going to probably turn over very rapidly because of the exposure to the dust and the dirt. If you live in an environmentally wonderful area where the air is extremely clean, your sensory neurons will live a little bit longer. But once lost, they're replaced by division among this population of basal cells. So this is what it actually looks like. These are two olfactory sensory neurons. There are many different olfactory sensory neurons that are present here. They're shown in blue. This is the surface of the epithelium. That bright green line you see at the top is where the mucus would be. The black up above it is where the nasal cavity is. You see a cell body, and you see an apical dendrite going up to the surface. You can't see the cilia at this level of magnification. And then you see another one next to it doing the same thing. I've mentioned a couple of times that the sensory neurons actually occur in many different subpopulations. So what you're looking at are two members of just one subpopulation in this particular slide. We've stained it with a procedure in which we're able to visualize just small subsets of cells. Now, if we take this same image and we look at it at a much higher level of magnification using electron microscopy, what we can see is much more detail about what it looks like. This is called scanning electron microscopy. What we do is we take a piece of tissue, we'll go into your nose, if you let us, and we'll take out a little piece of olfactory epithelium and we'll freeze it rock hard, minus 70 degrees centigrade. And then we'll basically hit it with a hammer. What happens when you do that? The tissue shatters into many different pieces. And what you're looking at here is a shattered piece of tissue. The reason that's helpful for us is it'll break across the depth of the tissue, and we can look inside at the surface and see what it looks like. This is the equivalent of looking with a magnifying glass at your olfactory epithelium at a magnification of around 15,000 times. It's called scanning electron microscopy. And you can see two cells here. One is the olfactory sensory neuron just here. Here's the cell body. Here's the apical dendrite. And you see just a little bit of the cilia up at the top. And this is the sustentacular or supporting cell that's just next to it. And what I'd like you to notice is the surface. This is the surface of the olfactory epithelium. And everything you see there that's very white are the cilia that I've been talking about. The cilia that increase the surface area of the sensory neuron and increase the probability of an odorant being detected when it comes in. And you'll see that in a bit more detail in the next slide. Okay, now we're looking at an even higher magnification. This bar is about 2 microns. The thickness of one of your fingernails is about 500 microns. So you can judge the size accordingly. This is the dendritic knob extending out onto the surface of the olfactory epithelium. And these are the cilia that are coming off. So they're out there waving like this. What we're going to find out is that some of these bumps that you see on the surface of the cilia are where the molecular detectors for the odorants are. So when an odorant comes in, a piece of that odorant is going to bind at a very small and restricted space on the surface of the cilia that you see here. If we look at that from a different perspective, let me go back. The next slide I'm going to show you is going to take this and we're going to cut right through this area. So we're going to look at a very thin section. We're going to ask about the density of these cilia within a space. This is what we've done here. This is called scanning electron transmission electron microscopy. So a dendritic knob is here, and all these little pinwheels that you see are the cilia. Remember, those are the ones that are responsible for detecting the odorants or interacting with the odorants when they come in. You can see how densely they're distributed at the surface of the olfactory epithelium, so that when an odorant does come in, the likelihood of it being able to move through this very dense field of cilia, of odor detectors, without interacting with the detector is very unlikely. So we have a dense distribution of cilia at the surface of the olfactory epithelium, all of them designed to detect and eventually interpret particular odors that may come in. So what happens? An odorant is going to interact with a particular site. This is the cilia shown here, and we're looking at it at higher magnification. When an odorant, we're showing it as a circle here, interacts with the molecular receptor, we'll look at the molecular receptor in a minute, there's going to be a change in the electrical property of the cell. We're just one bundle of electricity, right? If you press on your finger, there's an electrical event that occurs that takes that information, carries it up your arm and into your brain. Every time you see something, there's an electrical event that occurs. As you're listening to me, part of your ear is vibrating very rapidly in response to the sound pressures that are coming out of my mouth. And as a consequence, you're able to hear. Well, the same thing happens in the olfactory system. It's like a spark plug in a gasoline engine. When there's a spark, the piston moves and the information is carried onto the crankshaft and the car goes. Well, that's what happens here as well. The spark is the odor binding with the receptor that results in a change in the electrical properties of the cell, and if the change is sufficient, that information is then carried on to the central nervous system. We can talk about that more later if you want to, but you probably won't want to, so it's okay. So one young man asked me before the lecture started, why would you want to work in olfaction? Well, I mean, with opportunities like this, why wouldn't you want to work in olfaction? In my lab and others, we have opportunities available for both experimenters as well as subjects. So I know this is a little funny. Olfaction is a billion-dollar industry. Billion-dollar industry. How many of you have been to department stores and walked through the perfume section where they're selling perfumes for hundreds of dollars a bottle? Hundreds of dollars a bottle just so that you can smell a particular way. I hope everybody used a nice deodorant soap this morning. Maybe put on a little deodorant as well. Those things you put in the toilet bowl at home to make the toilet smell nice. The stuff that you spray in the house. Little things that hang from the rear view mirror. On and on and on and on and on. A multi-billion-dollar industry. And this is just one example of trying to determine whether a particular deodorant worked well. Also has disadvantages because it means that we're ignoring biological signals that help us to interact with each other. And we'll talk about that a little bit more in a few minutes. So I've told you that we have these populations of sensory neurons in the nose, and that they occur as different subpopulations. And those subpopulations are determined by the molecular receptor that each of those cells expresses. Now, if you look again at the somatosensory system, there's a nerve fiber in the tip of my index finger and in the tip of yours that responds specifically to high temperatures. There's another one that responds specifically to low temperatures. And even within that, there are some that are specific for high temperatures of a very specific range, and there are some that are specific for low temperatures within a specific range. There's another receptor in there that responds to very high frequency vibrations. It's called a Pacinian corpuscle. And there's another one that responds to low frequency vibrations. So these are called labeled lines. And what that means is a specific detector in your somatosensory system, in your visual system, or in your auditory system is going to respond to a very precise stimulus within your environment. The olfactory system does exactly the same thing. And what's extraordinary about it is there are more genes devoted to decoding odor information than are devoted to any other function in our body or any other function in our brain by a factor of at least 20. So I've been moving forward and I didn't even know it. So in the mouse, for example, there are over 1,000 odor receptors. This is called a dendrogram. We don't need to worry about the details of this. All you need to recognize is that within the dendrogram, if we look at this one little area, we see, 25 different odor receptors represented. So the detail shown here, not apparent here, just extrapolate and recognize that there are in the mouse over 1,000 different odor receptors. Isn't that extraordinary? The visual system only has really 4: red, green, blue, and kind of a night vision receptor. The auditory system really only has one kind of receptor. It's just that the tissue that is suspended in responds differently. Somatosensory only has about 6. Smell has over 1,000. It's incredible. Don't you think? Good. And in fact, I will mention that this was discovered by Dr. Linda Buck, working with Dr. Richard Axel. It was reported in 1991 in Science. And 2 years ago, Linda and her colleague Richard Axel received the Nobel Prize for it. And Linda is only one of 7 women Nobel laureates that have been recognized for their contributions in biology. Yeah, that is worth applauding. I have to come closer.
I said, does that infer that there's like 1,000 different types of cells, receptor cells?
Yes, 1,000 subpopulations. And each subpopulation will have, depending upon the animal that we're looking at, the mouse versus the human, anywhere from 2,000 to 10,000 cells that are within that population.
The parent cells that generate these cells, there's only one type of those?
We don't know. We don't know. We don't know what the mechanism is that results in a particular sensory neuron expressing the odor receptor mouse I7, for example. We don't know what regulates that process yet.
But the question is, if one of the subpopulation cells dies, does the basal cell replace that particular cell? In other words, that one that handles that kind of receptor, molecular receptor.
Yes. So if a mouse I7 cell is lost, when it's replaced with a cell that also expresses mouse I7. At least that's true during the bulk of our life. As we get older, in our 80s and 90s, that process becomes perturbed and it no longer works as well.
So any particular basal cell can make one of maybe 1,000 different types of receptors?
Right, so basal cells we call pluripotential because as far as we're able to determine, any of those 1,000 odor receptors that are expressed in the mouse can be made by any basal cell or stem cell. We don't know if they're multi-potential. We don't know whether they'll generate neurons that could replace a cortical cell, for example. But in the olfactory epithelium, they replace everything. Yeah, we don't know. It's a population of cells that seems to be quite independent from others. We don't know what it responds to. We don't understand it yet. We do know that the gray part up at the top, though, this is from the mouse, but these are also present in the human, are fish-like receptors. They respond to amino acids. The degree of conservation, that is the degree to which the odor receptors expressed in mice and humans and fish and other vertebrates are conserved or maintained through evolution is very high. And these are found exclusively, I mean, these are predominant in fish with not the tan ones shown at all in fish.
So they're kind of vestigial.
Yeah.
When you use the word express, is that the same as, how is the scientific use of the word express?
So when I say express, I mean that the molecular receptor, and think of the molecular receptor as a pocket. It's just going to be a pocket into which a molecule of a particular shape will insert, and we'll look at some of those molecules. When I say expressed, I mean that pocket has been inserted into the membrane of the cell. So it's on the surface, and it's available to interact with any stimuli that may come in. Humans have, relative to mice, about 350 odor receptors. Despite that, our sense of smell is much better than you think it is. You ignore most smells. You cover them up with deodorants, or you don't comment on them. Yeah. Or you may comment on them. You know, if your little brother was just in the bathroom and you went in there, you may have a very specific comment for your little brother. But one of the things that we don't do is we don't put our noses where the smells are. Smells are usually relatively heavy molecules, maybe a foot or so off the ground. And as opposed to dogs who are down on the ground with their nose in the dirt following a trail or a track, we tend not to do that. So if we're looking for a food source, for example. In terms of species recognition, friend versus foe, dogs put their noses where the smells are. And we find it socially unacceptable to do that. Yeah, I can't even imagine. But if we take different species, if we take dogs and mice and humans and carry out objective cognitive analyses of their ability to distinguish between different odors and their sensitivity to different odors, they're extraordinarily similar to each other. So dogs are better than us, mice are also better than us, but not as much better as you would have suspected. So I've been talking a little bit about molecules. I've been talking about odors. These are examples of odors. This is what fresh lime looks like. Sandalwood is going to look like this. A goaty mutton smell will be like this. This is urine, rose petals, cheesy, sweaty, violet, and licorice. And as you can see, some of these molecules appear relatively simple. Some of them appear much more complex. But on each of these molecules, there are what the olfactory people call determinants. There are methyl groups, there are oxygens, there are hydrogens, there are double bonds, there are single bonds, and there are carbon chains. And we think that each of those kinds of determinants will bind with a different odor receptor. And that's illustrated in the next slide. This is only to say that odor receptors have a molecular structure and that they're inserted into the membrane, as you see here, and that there is a pocket into which an odorant molecule or a piece of an odorant molecule can insert. So this is the fresh lime and the citrus. Let's say that the methyl group up here is equal to red and that the oxygen is equal to green. So this is the methyl group and this is the oxygen group. Now we can carry that across and recognize that in this particular example, we have 9 different kinds of receptors. We have red squares, blue, green, we have half circles, and we have triangles. And we can make the model in which the red square is going to bind with the red square receptor, the green triangle is going to bind with the green triangle receptor. Now, if we carry that one step farther, and now we say, all right, down here with the methyl group and the oxygen, the red square and the green triangle, that's what citrus smells like. That's what fresh lime smells like. So now when we stimulate or when we expose the organism to this particular combination, we're going to activate, again, the red square and the green diamond, and that will be then encoded as the scent of fresh lime or citrus. It's more complex than that because there are obviously more determinants or more features to this molecule than just the two I've picked out to look at. But that's where the term combinatorial code comes from. So an odorant, or what has a particular smell, is going to have a number of features, and each of those features will interact with a different kind of receptor, and then they'll combine, and together they'll give you the smell of lime or coffee or that pizza that I love so much. So what do we know so far? We know that there are sensory neurons in the nose, and that they have these ciliary processes on them, and that those cilia have molecular receptors that respond to different features of molecules. And that as those features are detected by those cells, that information then passes into the central nervous system. Yes? Uh-huh. Oh, down here at the bottom? Those would be other kinds of receptors. So in the mouse, there are going to be 1,000 different combinations of these. There are going to be red squares, blue squares, green squares, pink squares, and so on and so forth. So these are just other examples of different kinds of odorants. Yes?
Will the receptor release the odorant after some period of time?
Yes, and we think that those supporting or sustentacular cells may participate in that process. Also, the mucus that's in the nose, as most of you know, is continuously flowing, and it usually goes down your throat. You swallow it. The movement of the mucus will also help remove the odorants from the environment. Okay, so now let's go on and see what happens when we move farther into the brain. So this is what we've looked at so far: the sensory neurons, the cilia at the surface, the interactions of molecules with particular receptors. Now, I said that that information was going to move from the nose, from the nasal epithelium or the olfactory epithelium, centrally into the olfactory bulb, which is the first part of the brain that we can look at, where they're going to make connections with other neurons that in these spherical regions, and they are spherical, as you see here, that are called glomeruli. And then those neurons, in turn, will take that information and distribute it to many other parts of the central nervous system, where it's used to regulate feeding behavior, aggressive behavior, sexual behavior, and often just a sense of well-being. So let's take a look at the olfactory bulb a little more closely and what happens. In the human, I do have some brains with me, by the way, that I brought in. And afterwards, when we're done, if you'd like to come and look at them, and I'll give you a quick little tour, and I've got some gloves as well. So this is the ventral view of the brain. I'm looking up like this. This is part of the brainstem. This is where the spinal cord would come off. This is cerebellum. It helps modulate movement. And what we're talking about is this little tiny structure up here and right there. This is the olfactory bulb. This is the axons from the olfactory bulb going back to other cortical regions that are also responsible for processing odor information. So our axons from our olfactory epithelium are going to go into this structure and this structure. So we're back at the epithelium once again with the slide that I showed you before. And you see these very thin processes that are coming off the bottom part of each of these cells. Those are the axons. They move back to the olfactory bulb, and you see the left olfactory bulb and the right olfactory bulb in the image on the right. And the green processes that you see relative to the red are the axons coming from all of the cells that express the same odor receptor that these two are expressing. Happens to be an odor receptor called M71. And these axons will continue to move across the surface of the olfactory bulb until they find the right glomerulus into which they want to go and make synapses or contact the other cells that will take that information to other parts of the brain. That's seen perhaps a little more clearly here. So this is from the mouse. It's called a mid-sagittal section. We've cut the head right down the middle so that we can look at the olfactory bulb. We're seeing the olfactory epithelium here and the olfactory bulb here. Now, rather than staining the cells that we're interested in, cells of a particular subpopulation that express a particular odor receptor, this happens to be odor receptor P2, rather than looking at just a couple of them, we're looking at all of them. You can see the little dots in the olfactory epithelium, and then you see these larger blue lines. Those are the axons coming together as they're moving from the olfactory epithelium back to the olfactory bulb. And what I hope you'll recognize is that within the nose, those cells are all over the place. They're very broadly distributed. But as those axons go back to the olfactory bulb, they all terminate in one little spot in what's called the medial glomerulus for the P2 odor receptor. So all of the cells that express the same odor receptor, that is, all of the cells that respond to the red square or all of the cells that respond to the green diamond and so on and so forth for different kinds of determinants, will go back to just a single glomerulus within the olfactory bulb. And there are many glomeruli. Now we're looking at the surface of the olfactory bulb. The nose is down here at the bottom. The rest of the brain is back here. This is the left olfactory bulb. So if we turned it upside down, you'd see it oriented more normally. And each of these dots that you see is a glomerulus. There are about 1,800 of them in the mouse. So there are two glomeruli, 2 points at which the axons expressing the same odor receptor, molecularly defined receptor, converge in the olfactory bulb. So this is like a map, right? This is a methyl group. This is maybe a carbon group. This is an oxygen group, and so on.
You said that we have like 1,000 receptors, right?
Yes.
We have 1,800 glomeruli. So some are going to more than one?
It's biology. Some go to 2, yes. And there is some variability. Some go to just 1, many go to 2.
And these two are the same, or they are different and they are like combinatorial?
It's a great question. This is the medial part of the olfactory bulb. This is the lateral part of the olfactory bulb. And each odor receptor, or each subpopulation, goes to a medial, by and large, goes to a medial glomerulus and a lateral glomerulus. And we don't know whether they're functionally different or not. Yes?
Is the signaling of all the different groups identical because they all go to their determinants?
It seems to be equivalent. So the sensitivity, the ability of a particular molecule to elicit an electrical event in a particular sensory neuron seems to be equivalent across the full population of odor receptors. So let's review. The olfactory bulb is up at the top. The olfactory epithelium is down at the bottom. We have this enormous population of sensory neurons. All of the cells that express the red receptor, whatever that may be, converge onto a particular glomerulus, or 2 glomeruli in the olfactory bulb. And from there, what are called projection neurons take that information out to other parts of the central nervous system, where it's used to regulate things such as appetite, sexual behavior, and so on. All of the green sensory neurons, the ones that express a different odor receptor, go to a different glomerulus, and so on and so forth for every odor receptor that's expressed in your olfactory epithelium. So for us, there would be approximately 350 different colors that would be represented in the epithelium. In the mouse, there would be approximately 1,000 different colors that are represented in the epithelium. Now yes?
Is the strength of the smell dependent on how many cells are activated?
Yes.
Each cell is on or off, but the more on ones you get.
Exactly. So the strength of a stimulus that is pressing on your finger is determined by how great the pressure is. And in turn, the strength of an odor is determined by how many sensory neurons are activated.
Do the odorants work by interfering with the attachment of the odorant to the cilia, or do they work like white noise by overcoming with such a strong scent that you have other scents that sort of elude it?
I'm sorry, could you ask that again?
I imagine that the way white noise works is that it gives you such a broad stimulation that you don't hear the siren outside your window when you're trying to sleep. Do the deodorants do the same thing, or do they interfere with the interaction of the odorant with the ciliary receptors?
So most of the odorants that we know about are blocking the odor source, or they're masking the odor source in some way. And not, we don't believe that they're acting directly at the receptor level. We think they're acting on the odorant molecules before they get into the nose. Yes?
So there were 1 to 5 million receptors, sensory receptors. If I have 5 million, I can smell better?
Not necessarily. It's possible, but it's not assured. Dogs have more sensory neurons than we do, and they do smell better, but it's probably because they have more molecular receptors and not just more sensory neurons.
What if the tank is wrong one of those neurons when it doesn't like smell different? Just because of the wrong one, or is it
Yes, that's a great question. So what happens when a green odorant activates a red receptor cell? It doesn't happen very often, but when it does happen, you can misinterpret the smell and think it's something else. The analogy would be, if I come along and punch you in the eye, I won't do that. But if I did, you would see light. There would be a light flash that would happen. It's not because your retina or the receptor cells in your eye were properly stimulated. It's because of the physical deformation that occurs. So the stimulus is inappropriate, but there's still a sensory-like event that occurs. We can have similar effects in the olfactory system.
I guess a related question is when one of these cells dies, is it understood what process to get new cells to reconnect to the right?
Boy, you want to come work in my lab? No, we don't understand that. It's an intense area of study right now. It's important not only for the olfactory system, but it's important for people that have had strokes, traumatic brain injury from car accidents, spinal cord injury, and so on and so forth. So there are a lot of labs that are interested in that very problem and looking at it not just in the olfactory system, but elsewhere in the brain as well.
How do you learn to associate certain chemical combinations with certain things? Like, how do you learn to associate the smell of pizza with pizza?
That's a hard question. And I'm going to skirt it a little bit by giving you a couple of examples. We know that if your mother ate a lot of garlic when she was carrying you, when she was pregnant with you, you're going to like garlic. Now, your postnatal experience as you're growing up, your mother probably still likes garlic, and there's going to be a lot of it in the cooking. But we know that in utero experience, when you're floating around in the amniotic fluid of the womb, that there are smells and tastes that can pass the barriers that are found, and they will influence your preferences. So we can take a rat, and we can inject the odor of peppermint into its amniotic fluid. After the rat is born, we cross-foster it. We put it with another mother that has never seen, doesn't know anything about peppermint. She'll raise up that rat litter, and they'll be just fine and normal. And then later, 6 months old, a year old, we take those rats or those mice and we put them in a situation in which we say, would you rather be around peppermint or would you rather be around normal cage smells or spearmint or some other odor? Besides, normal cage smells are awful, so pizza smells. They'll go with the peppermint. They'll go with the peppermint every time. They'll also preferentially mate with another female or with a female that's scented with peppermint as well. So we know these associations come on very early in the embryo. Beyond that, I think it's a positive experience. If you go out with your friends, you have a great time, and it's the first time you've had a combination pizza, but everybody's having a good time, and you're laughing. It's reinforcing. You like that experience, and you associate that with the food. You can contrast that with going out, and maybe a lot of you have not yet gone out and had a pizza and gotten really sick afterwards. Now it's an aversive relationship. And now when you smell that combination pizza, it's disgusting. You don't want to go near it. My sister fixed eggs for me when I was little. And I got very sick for who knows what reason. I cannot stand the smell of eggs. I don't want to be anywhere around them. And it's because I got sick just once, called one trial learning. It's a survival mechanism.
Smell, memory, and recognition all get tied together.
Yeah.
At least in human experience.
Right. So there's a famous author from the turn of the century, a French philosopher and author, Marcel Proust, who wrote a book, a huge volume, called Remembrance of Things Past. And what he talks about is the power of a particular sensory experience, an odor in this case, to elicit a flood of memories. So as an adult, he was having a cookie with some tea in it, and it elicited in him this incredible warmth and remembrance of the events that had taken place when he was a child, and he was having tea and a cookie with his mother. Hadn't thought of it in decades, years and years and years, but suddenly that smell stimulated that memory, and it just came flooding back. Olfaction seems to be particularly strong in that way. You can go to your grandparents' house or your home where you haven't been in years and years and years, and you walk in and you know you're home. You know you're home immediately from the smell.
I'm not trying to give a moral lesson to the young people, but it seems that smoking increases your sensitivity of your olfaction.
Sure. So I pointed out already that the olfactory, the sensory neurons in the nose are very delicate. And can you think of anything nastier? Well, I can think of other nastier things. But smoke is certainly among the nasty things to put in your nose. And if you keep killing these cells again and again and again unnecessarily, you're going to have a decrease in your sense of smell. We know that people that have been heavy smokers and then stopped, that within a period of time that we think is consistent or appropriate for reconstitution of the olfactory epithelium with sensory neurons, their sense of smell will return, or it will improve dramatically at the very least. So smoking is a toxin. You know, you take a drag on a cigarette and you blow it out through your nose. You're going to be exposing those sensory neurons. You're going to be killing them. And you're going to be killing them so fast that sense of smell will decline. Yeah. Yeah, but your parents might not like it if we gave you that virus and got you really sick. But in fact, we do experiments like that. We take a drug called lithium chloride, and we'll expose an animal to a smell, and then we inject them with lithium. So the question was, could we do an experiment in which we expose someone, a kid, to the smell of smoke, and then give them a virus that's going to make them really sick, and then make them make the smoke aversive to them to keep them from smoking in the future. And in fact, we do experiments like that on a regular basis with rats, not with smoke, but with other things. We'll expose them to an odorant, then we inject them with a drug called lithium chloride, which is the equivalent of the worst stomach ache you've ever had. And then we ask them later on, now are you willing to be exposed to this particular odor that you previously liked, and they don't want to have anything to do with it. It's like with me and the eggs. So we can, in fact, do that. 3 hours, right?
If you were studying for a test or something and you were smelling a strong scent while you were doing it, and then when you're taking the test or whatever it was, you smelled that scent again, would it help you remember?
It's called state-dependent learning. And what happens is that there's evidence that individuals do better when the testing environment, the classroom, is the same as the environment in which they're studying. And in fact, on the front page of the New York Times several weeks ago, there was an article about that very phenomenon in which individuals that studied around the smell of roses and then were exposed to the scent of roses while they were sleeping, showed evidence of having a better memory the next day for the material that they had studied, math or literature, whatever it might have been. But how that works is not well understood. God, how awful. What happens if there are people that have never smelled pizza before? Can you imagine anything terrible?
And then one person really likes the smell, and then one person doesn't.
So that happens all the time. You know, kids that grow up in different cultures. My background is Swedish, among the Swedes during holiday seasons. There's a fish that's served that's called lutefisk that is disgusting for most people, but I grew up with it, so I happen to think it's fabulous. And that happens a lot. There's certainly people. My wife doesn't like pizza. I mean, how awful is this? My favorite food in the world, and my wife doesn't like it. So I don't get to have it as often as I like. But you're absolutely right. You know, there are people that can like and dislike based upon their early experience. Let me go quickly through a couple of slides, because at the end, I'd still like to do this experiment, which means jelly beans, which is a good thing. I also have these human brains up here for you all to come down and look at if you'd like to. Yeah, yeah. I think so, yeah. I hope so. I'll tell you a little bit more about the brains before we get there. So we can make some predictions about the way in which odor information is going to be represented. That the cells that detect green odorants will go to the green glomerulus, and the cells that detect red odorants will go to the red glomerulus. And we can carry that on with the model that we established earlier. Remember, the methyl group is going to be red and the oxygen is going to be green. So it's going to activate a very specific subset of receptors. And because all of the receptors that express the same molecular odor receptor go to the same glomerulus in the olfactory bulb, if we expose a rat or a mouse to this particular molecule, and we know that this is the code, we can now predict with certainty that this glomerulus and this glomerulus, or these two regions of the olfactory bulb, will be the ones that will show functional activity following exposure to this particular odorant. So, you know, can we test that? Can we find out if that's really true? We can do that in mice. So this is a little hard to look at, but we're looking down at the surface of the olfactory bulb. These black lines are the blood supply. This is a live animal. And when functional activity occurs, it's going to appear as this very black area. Through image processing, we can determine that the blackest areas are indicated here in red. And we can then carry that image processing further and identify that this glomerulus, and this glomerulus are the three glomeruli in this particular animal that responded to the odorant that the animal was presented with. So it wasn't lime. It was an odorant called amyl acetate that smells like bananas. But these were the three glomeruli that were active. So the model that we had, in which a particular subset of sensory neurons in the nose will be active with exposure to a particular odorant, will then result in the activation or functional activity in a particular subset of glomeruli in the olfactory bulb, seems to hold true. These other glomeruli are inactive. Yes?
How is activation detected in order to get the
So there are lots of ways to do it. We can look at changes in the levels of ions, calcium, sodium, potassium. The other thing that happens is when your brain is active, the cells that are there will swell and shrink a little bit. And when they swell and shrink, the degree to which a light reflects off the surface of the brain changes. And that's what this is. This is a change in reflectance that we have shown in the past and others have shown in the past is associated with functional activity. You can also use something like functional MRI that you may have heard of in the past. MRI is usually used in a diagnostic way, but you can also look at functional activity in the human brain using MRI as well.
What are the green stars for?
Oh, they're just to identify landmarks so that as you go from image to image to image and you're processing it through the computer, you can coordinate where things are. So if you continue with this model, doing that kind of thing, you know, the C3 through the C7 down here, what those are specifically is not important, but only to know that they're different determinants or different components of odorant molecules. And what we're showing you here is that different odorants will activate different regions of the olfactory bulb. So there's a map on the olfactory bulb. Different kinds of odorants will activate different regions. Subsets of sensory neurons respond to very specific components or pieces, parts of molecules. They put those together and then they all project into the olfactory bulb where they activate subpopulations of glomeruli. And that in turn is converted into a map on the surface of the olfactory bulb. And with experience and by looking at lots of different odorants, we can look at a map like this and we can tell you what the odorant was. So in the same way that we can look at a map of your finger and the surface of your brain, which we do very effectively, we can now look at the surface of the olfactory bulb as well and tell you what kind of odorant, or at least what class of odorant, you may have been exposed to as a result, or that results in that particular pattern of activity. Now, I'm almost done, I promise. Another hour and a half will be there. I've mentioned to you that odorants affect behavior. I told you that in utero that you had odor experiences that influenced preferences and things you liked after you were born. In utero, you're exposed to the amniotic fluid that you're floating in the womb. And when you're first born, your initial suckling, your initial attachment to your mother's nipple to initiate suckling behavior was determined by an olfactory cue. If we take the breast pads from two mothers that gave birth at the same time, same day of the birth, and we take the two infants from mother A and mother B and expose them to the breast pads from mother A and mother B, and ask which one they oriented to, or which one they prefer, baby A from mother A orients preferentially to breast pad A, and baby B orients preferentially to breast pad B from the mother. If we change the amniotic fluid, if we change the odors that are there, not in humans, but in animals, now they'll no longer suckle or initiate suckling on a normal breast, that is one that is unscented. They'll only go for the scented breast. So olfaction, less so among humans now, because the mothers, can obviously control the infant and their behavior a bit more precisely, but among other species, dogs, rats, cats, mice, so on and so forth, mammalian species, suckling is a critical determinant for survival. You don't smell, you don't eat, and you die. So at the very least, an important determinant. Among rodent populations, mice and rats, olfaction is a critical determinant in mate selection. Right? If you smell right, I'll mate with you. If you don't smell right, I won't mate with you. Now, I know what you're all thinking. Oh, I'm a human. I'm above that. I'm more sophisticated than that. But in fact, when we look at isolated breeding populations of humans, places like Pitcairn Island, for example, where the selection of breeding opportunities or selection of mating opportunities, marriages, I guess, is more limited, what you can recognize is that there is an odor specificity that occurs there as well. We have a number of markers that we can use to demonstrate that. So odor selection is a determinant for preference. And some of you that are older, perhaps you've been dating already, and you can recognize that, I don't want to be around that guy, versus, oh, well, that's okay. So, it influences your daily behavior. It influences your food preference. Yeah, so while they're passing out jelly beans, take several jelly beans from the bag. Do not eat them yet. Don't put them in your ears and don't put them up your nose. Just hold on to them. As soon as everybody has 3 or 4 or 5, 6 jelly beans, then I'll describe the experiment to you. I haven't talked about that. No, parallel system called the accessory olfactory system that may or may not be present in humans. It's not clear. Are there other questions I can do right now while the jelly beans are being passed out? Yes.
Keep mentioning is the science used in things relating to like rat control, trying to rat extermination, or pest extermination?
Yeah. So it is used in pest control, and I think even more importantly, some of you can you guys still hear me? Yeah. Yeah. Okay. So some of you may have heard John Carlson's talk last week, and I think the work that John is doing on looking at mechanisms of olfaction in mosquitoes that are vectors for malaria is perhaps the best example and one of the most profound examples of how people are interested in understanding how olfaction works in order to control what is otherwise a devastating disease. It's also true that if we could control the number of crops, the grains, the food that is destroyed by pests in the United States, if we could control that through olfaction, hunger would be obliterated. It would be gone. Yeah?
Do the stem cells age as the person ages and it doesn't function as well? That's why the sense of smell is less?
So we've just started a new program project with Linda Buck, the one that got the Nobel Prize, to ask that very question. We don't know why olfaction in humans as they get older goes into decline. I'm sorry. So the question was, does something happen to the stem cell population in the aged that results in decline in olfactory function? And can we understand how stem cells work by looking at that? And the answer to the question is, we think so and yes. We've just started that project with Linda Buck to take a look at that very question. What happens? What are the underlying mechanisms that are responsible for the decline in olfaction that occurs in the aged? And one very real possibility is that the reproduction of new cells by the stem cell population decreases.
How fast does the neuroepithelium regenerate? How fast?
So how fast does the neuroepithelium or olfactory epithelium regenerate? And it takes about, if you destroy the entire thing, which we can do chemically, it takes about 6 weeks for the entire epithelium to be reconstituted and connect with the olfactory bulb.
How does the common cold virus affect the olfactory?
Very badly. I mean, many of you have found a diminished sense of smell after a bad viral infection of your olfactory system, and it's slow to recover.
In that experiment we just described, does the population of the receptor cells come out the same as the original one?
If there's traumatic injury, you can sometimes have the formation of a scar, and then the organization of the system is destroyed. The distribution within the epithelium stays the same, but with a scar, it sometimes means that their ability to innervate centrally, the olfactory bulb, is decreased. No, she was first. Again? These? Oh, the cloth is wet on top of them. Otherwise, normally the brain in the skull is suspended in a large vat of fluid that's called cerebrospinal fluid. So the brain is actually floating all the time. I mean, that certainly contributes. So there's a lot of mucus production, and the sinuses get clogged, and they're pumping out junk. But our experiment that I'm about to do will bear on that.
Can you explain a comment on beagles being used for drug detection?
They could use you for drug detection. It's just a question of training. It takes 2 or 3 years to train a dog to detect drugs or to detect plant products. If you've seen them at the international arrivals at JFK, JFK or Dulles, for example, you see the beagles running around. Beagles are good because they're small, they're amicable. Everybody likes beagles, as opposed to Doberman Pinschers, which produce a different reaction. So it's just a convenient animal. But it takes about 2 to 3 years to train one to detect a particular substance and to behave in the appropriate way. And even then, that training has to be continually reinforced.
Are there any odors that chemically produce that are known to cause brain cancers or learning disabilities?
Sure. So there are lots of odorants that have clinical consequences associated with them. And as you pointed out, some odorants are irritants. If you smell them, it burns your nose. That doesn't have really anything to do with the sense of smell. It has to do with other sensory systems in the nose, like the trigeminal, for example, that respond to somatosensory information, and not the olfactory system per se.
Why don't we hold off on questions and
Okay.
ask me about the speaker at the end and try the experiment?
All right. All right. So let me first tell you what you're going to do, and then we'll walk through and actually do it. All right. So don't do anything yet. What I'm going to have you do. Well, all right. When you have that combination pizza that you really love, or that I really love, how many think that it's your mouth that's telling you what you're tasting? Partly? Okay, so partly at least. Okay, so let's test that. What you're going to do, don't do it yet, is you're going to pinch your nose very tightly. Don't do it yet. Pinch your nose very tightly so that no air is moving through, so you can't breathe through your nose. While your nose is pinched, you're going to put the jelly beans in your mouth, keeping your nose pinched while you do it. Put the jelly beans in. You're going to chew, and just chew for a few seconds, and I'll tell you when to let your nose go. Okay, so pinch the noses. All of them at once, yeah. Go for the full experience, the combination. All right, pinch the nose has got to be pinched before the jelly beans go in. All right? Nose pinch, jelly beans in, chew, chew, chew. Some texture, some sweetness that's coming off of the tongue. Okay, let go of your noses. Works every time. So what happens is this. This is your mouth. This is your tongue. Your tongue responds to sweet, sour, salty, and bitter, and that's it. Doesn't give you flavor. Doesn't give you that cherry flavor, blueberry flavor, island punch, I think was the other one that I had in there. What happens is when the nasal cavity is patent, when you don't have a cold, or when your nose is open normally, air from your mouth goes this way. It's called the retronasal pathway. It goes from your mouth very rapidly up into your olfactory epithelium, stimulates the olfactory sensory neurons, and you have the experience of cherry combination pizzas. If you're a wine drinker, you see the wine people smelling all the time. It's to activate these areas up here. When we block the nose, we block the flow of air up there, and as a consequence, you block perception. If you're old enough, when you were little, maybe your mother pinched your nose while you had to take some medicine that didn't taste good. She pinched your nose so you couldn't taste it. So that's what you've experienced today. And thanks very much.